Hello, welcome everyone. I'm Joshua Stein, Project Manager at the International Wheat Genome Sequencing Consortium, and it's my pleasure to serve as moderator for today's monthly webinar series. So welcome everyone. I'm going to pause for a moment and let more people join. And today we're joined by Rimsha Ashraf, uh, assistant professor with the Swedish University of Agricultural Sciences. And before we continue on to Rimsha's presentation, I'd like to provide an overview of the IWGSC. We are an international nonprofit consortium with 3,450 general members from 943 institutes and companies and located in 76 countries. And I'd also like to take this time to acknowledge sponsors of the IWGSC who make this webinar series possible. So please, let's all thank Arbor Biosciences, BASF, CIMMYT, Florimont Desprez, INRA E, Kansas Wheat, RAGT, and Syngenta. So thank you again to our sponsors. So as many of you may know, the IWGSC has long been involved in genomics, providing genomic resources to the wheat community. Our current vision 2.0 is to enhance breeding through an increased understanding of the molecular basis of traits and their allelic diversity. And I'm very happy to mention a new project, a new initiative that works towards this vision, and that is the IWGSC Wheat Diversity Project that's now in its sixth month. And so the goal of this project is to preserve, discover, and mobilize genetic resources by sequencing landrace genomes that encompass the worldwide diversity of bread wheat. So this includes platinum level assemblies of 12 landrace genomes, as well as an update to Chinese Spring in order to facilitate gene annotation. Transcriptome data will be collected from multiple developmental stages and tissues for each of the genomes in this study. And then finally, in order to make the data useful to the community, we plan to catalog all the genetic diversity using a practical haplotype graph. And so please stay tuned as we continue on this project, which is ongoing. I do want to mention upcoming webinars. So on October 17th, we'll have Yves Desmet from IWGSC Center for Plant Systems Biology, and he'll be discussing comparative wheat phosphoproteome profiling. So please join us in October for that. And then finally, I want to mention upcoming workshops. So this September 19th will be the IWGSC workshop at the Plant and Animal Genome Conference in Australia, Perth. And I'm very happy to be organizing this workshop and will be chairing it. We have 5 great speakers that will be attending. I look forward to meeting anyone who will be attending who's also on this webinar. I look forward to meeting you all. And then a new opportunity, 2 IWGSC workshops will be held at the Plant and Animal Genome Conference in San Diego this coming January. So very soon there'll be a call for abstracts. So please keep an eye out for that so that you may attend. And this is going to be a very special Plant and Animal Genome Conference for the IWGSC because we'll be celebrating the 20th anniversary of the formation of the IWGSC. So we hope to see many of you there and help us celebrate these remarkable accomplishments. Before I begin, I just want to give you a little bit of a tutorial on the interface for this. So the webinar is being recorded, and it'll appear on the IWGSC YouTube channel, so stay tuned for that. The presentation will be followed by a question and answer period. During the presentation, you may click on the Q&A button and leave a question. The questions will be prioritized by people's upvoting. At the end of the presentation, I will ask the questions of Rimsha. And if time allows, we'll get through all the questions, but if time does not allow, she has agreed to answer the questions in writing, and we will distribute those. There's also a chat panel. This is useful for communicating. The director of this webinar is in the background and will be sending chats. Isabel, she'll be sending chats with information about this webinar, such as handouts, etc. So once again, please, our mailing list is down at the bottom if you'd like to sign up for the IWGSC newsletter. Okay, finally, let's get to the main course of this webinar. We have today Rimsha Ashraf from the Swedish University of Agricultural Sciences in Sweden. And today she will be talking about her research on the identification, characterization, and combination of novel resistance genes against rust in wheat. So thank you again for joining, and welcome, Rimsha.
Thank you, Josh. First of all, thank you very much for you guys, as well to IWGSC, who gave me opportunity to talk about my research. Topic of my today webinar is identification, characterization, and combination of novel resistance genes against rust in wheat. I recently did my PhD from Swedish University of Agriculture Sciences. So before moving forward, I would like to give you the outline of today's webinar. First, we will talk about background, aims and objectives of my research, potential of wheat alien introgression lines, and then role of rye chromosomes against stripe rust, combination of stem rust and stripe rust resistance genes, conclusions, future directions of my research, and then acknowledgments. So first I would like to share with you about my background. I'm basically from Pakistan. I did my master in the subject of botany from Quaid-e-Azam University, Islamabad. And then I did my master of philosophy in plants genetics and genomics from the same university. Then I moved to Sweden, and recently I finished my PhD in the subject of biology, specialization in plant breeding from Swedish University of Agricultural Sciences. As we all know that wheat is one of the major crops in terms of importance, as well in terms of the food security after rice and maize. So, but sadly, a serial killer called wheat rust fungi ravaged every year approximately 15 million tons of wheat, which is equal to $3 billion US. And if we use all of this wheat, so it's enough to bake 25.5 billion loaves of bread, which can be used by 425 million people. That's quite a big number. It's equal to half of the population of Europe. So 3 types of rust fungi are there. As you can see in this picture, these yellow pustules on the wheat plant. It's called yellow stripe rust, which is caused by Puccinia striiformis FSP tritici. And then the temperature required is 2 to 15 degrees Celsius. That means it's a little bit moist temperature is required to start the infection. And the dominant races are warrior and graminis in the Europe. And the second is black or stem rust, which is caused by Puccinia graminis FSP tritici. These orange to red pustules on the wheat plant, as you can see in the picture, are stem rust, and the temperature is a little bit warmer than the stripe rust, 15 to 30 degrees Celsius, and these are the dominant races in the Europe. And the third is orange and the third is the brown or the leaf rust, orange to brown spores. The temperature required, it's also a little bit moist than the stem rust. So it's 15 to 22 degrees Celsius. So my major focus during my PhD was on the stripe rust. As well, I did some part on the stem rust too. So what make it actually most complicated to handle for the breeders as well for the farmers is it requires the 2 living hosts to complete its life cycle. First, it need a primary host, wheat, and the second is a barberry, the secondary host to complete its sexual and asexual life cycle. As you can see in this picture, these ball-like structures, which having spikes, these are tearing throughout the way of the epidermis of the leaf. They are ready to infect to the barberry plant in the next season. Similarly, on the secondary host, on the barberries, these tube-like structures having millions of spores, which are ready to reinfect the wheat plant in the next season. So in that way, they complete their life cycle, which actually make it most complicated to handle this disease. However, there are several of the legislations and restrictions to remove this barberry plant. Here is a picture and a poster of one of the successful campaign which occurred in North America from 1918 to 1980. Similarly, the same campaign also occurred in the Western Europe, like in Denmark, as well in Norway and Sweden. Sadly, different reports, they shows the infection of the stem rust in different parts of the world, like in one of the major epidemic which occurred in Italy, in Sicily, and then in several parts of the Western Europe. In Sweden, 2017, it was also detected. And then in UK, and most recently in South Asia, UG99 was also detected. And if we look to the other side of another disease of stripe rust, which is also spreading rapidly across the globe, as you can see in South America and in Australia, it's PST-S10 and PST-S13. These are common. So if you go to Europe, so you can also see PST-S10 and S13. These are actually the races of the stripe rust. So you can see how quickly it's spreading around the globe. This is a picture of the wheat field in Sweden during 2011, and this yellow part is not a paint. It's actually the stripe rust. If we zoom into it, so it actually looks like these yellow pustules on the wheat leaf. So we have a problem. Now the question is how we need to respond once the disease becomes established. So one suggestion is agronomic practices. Of course, we can do it. And the other is uses of the modern fungicides. We can also do it. But European Union Farm to Fork strategy actually aims to reduce the pesticide use and risk by 30% by 2030. So we really need to focus on resistance breeding. And for resistance breeding, what is actually the reason to choose this strategy is because it's environmental friendly and it's economically effective approach. And for this purpose, in our study, we use the rye. The reason why we are using rye, because it's wild relative to wheat. It gives a durable source of resistance against biotic as well, abiotic stresses. And several of the researchers, they have used the rye chromosomes for resistance breeding. And these are the name of the rye chromosomes, 1R, 2R until 7R. So the aims and objectives of my research was to identify the new sources for stripe rust resistance within wheat alien introgression lines as well wheat rye introgression lines. And then characterization of those particular stripe rust resistance genes by doing physical mapping and developing the Cas markers for those targeted genes. And then incorporation of this small rye chromatin within the wheat genome. And the last is combination of stripe rust and stem rust into a single adapted cultivar. So the material which we have used in our study, it's a large set of wheat alien introgression lines. It has wheat rye introgression lines as well wheat limus and wheat thinopyrum. So the material was actually developed by a late professor, Adolf Mirker, so special thanks to him, and it's a great opportunity to work with this material. The method which we used for these large number of lines are, we have checked these lines for agronomic performances, for diseases, and for pest screening, as well for allelopathic potential. And then we checked those lines for end-use quality analysis, for grain samples for micro and macronutrient concentration, as well for the protein composition. So we figure out that wheat rye introgression lines, they were highly resistant against the stripe rust viruses. And there were also some set of lines which were resistant towards the Hessian fly. And some lines, they also shows high levels of zinc and iron and very low cadmium concentration, which is really good for human health. And some lines also shows large variation in bread making quality. So overall, those set of wheat alien introgression lines actually improve the production, resistance, and the quality of the wheat. So we target wheat rye introgression lines, which were highly resistant for the stripe rust disease for our further analysis. So this paper has been actually published, so you guys can also check. So by targeting those wheat rye introgression lines, we did another study by using this rye chromosome for stripe rust resistance gene. So we have these wheat rye lines, which has 4R, 5R, and 6R rye chromosomes. And the idea was to identify the stripe rust or yellow rust resistance genes. And we use phenotype and genotype analysis to get the confirmation of our newly identified genes. In this picture, this is our parental line, SLU 126. So we checked this wheat rye introgression line against, these are actually set of lines, not only one line. It has 6 lines, which are called, we call it like sister lines because they have more or less similar genome composition in terms of rye introgression. So we checked this line approximately 30 yellow rust races from different races, like from UK, from Denmark, from Tajikistan, Turkey, Sweden, so different races, from different races, and we figure out that these lines are showing strong resistance. Then we move to the cytogenetic analysis, and we figure out that these wheat rye interrogation lines has 4R, 5R, and 6R rye chromosomes. So this is the picture of a chromosome. Like as you can see, this green dye actually shows the presence of the rye chromosomes. Before moving forward in our study, we need to consider the role of the pairing homologous loci, which is present on the 5B long arm of the wheat chromosome. Its function is actually it inhibits the homologous recombination. Means like when you are going to cross wheat with the rye or with any other or with any of the other species. So this is the beauty of the wheat genome, that it prevents this crossing, so it makes it more stable. So for this purpose, we need to switch off this pairing homologous loci. And in that case, this Chinese spring PH1B mutant, which actually don't have this PH1B, it really helps us to get the right chromosome within the wheat genome. So we crossed our line SLU126 with this Chinese spring PH1B mutant, and then we allowed it selfing until BC1F4 generation, and other set of lines we crossed with the adapted cultivars and allow it selfing until BC2F3. So throughout this process, we actually identify the recombinants in the early generation when we crossed with the Chinese spring PH1B mutant to get the confirmation of the absence of this PH1B loci. So we confirmed that those lines don't have. And then we did the sibling analysis against these 5 stripe rust races in each generation to get the confirmation of the resistance pattern in each generation. Apart from this, we also checked with the SSR markers to detect the rye chromosome in each generation, either it is present or absent, but it was not working. Sometimes it was like switch on and switch off, so they were not reliable. So at that time, we were unable to get a clear picture, either this resistance is from which chromosome. Of course, it was from the rye, but we were not sure that from which chromosome the resistance is coming. In this picture, as you can see, this is the parental line SLU126 and Chinese spring PH1B mutant, and the other Chinese spring as well SLU126. So when we check with the mix of these 2 stripe rust races, we figure out that in the next generation after selfing, this SLU126 is also resistant, and the others, they are susceptible in the next generation too. So here, this is a picture of some of the lines in the later generation. Like this line 245 in BC1F3, it is resistant. And the other line, 180, it is segregating. As you can see, 2 are susceptible and 2 are resistant. However, in the next generation, this line, 245, it's also resistant in the next generation. So similarly, line 180, it is also segregating in the next generation, which gives a signal that there are resistance genes which are actually coming from the right chromosome. Since we already had a clear picture of our parental line, which we actually crossed with the Chinese spring PH1B mutant, that it has 4R, 5R, 6R, but in the later generation, we were unable to detect that by using SSR markers, that which chromosome is actually responsible for the resistance. So we checked our resistant lines with by using the cytogenetic analysis, FISH and GISH, and we confirmed that there is from cytogenetic analysis, we got this picture that there is no Y chromosome, but the resistance is there. And then we just stopped at that point that what we have to do. So we moved to the GBS analysis. In the GBS analysis, we did with the parental lines, SLO 1 to 6, as well with the Chinese spring. And then we also did the GBS in the later generation for those lines which were strongly resistant with the different races. And when we got the data from the GBS, we aligned it with the wheat and the rye reference genome, and we figure out that in our case, we have 10,675 number of high quality SNPs after filtering, which are coming from 6R Y chromosomes. Then we use the NLR parser software, since we were targeting the NLR genes, which are really important, which actually activate the immune system of the host plant. So we figure out by collaborating with IPK and John Innes that in our case, in this line, SLU 1 to 6, we have 26 NLR genes which are coming from 6R Y chromosomes. In total, NLR genes which are coming from 6R Y chromosomes are 184, but we were lucky enough to have the quite high number, in our case 26. So we developed the markers, and then we can, as you can see, this is a physical map of SLU 1 to 6, and then we confirm that 26 they are present in our lines, but we were not sure that these all are for stripe rust, or they may be for different diseases. When we developed the CASP markers, so we identified 2 different genes which are present on different positions. So there's a big difference on their position. So 3 markers, they are actually amplifying one resistance gene, and then one of the markers is amplifying another resistance genes. So this is a picture of this is actually the picture of our CASP analysis, and it shows that the translocation is actually cryptic. That means which could not be detected by the cytogenetic analysis, since it was a very small translocation. So for this, the GBS is actually useful in our case. So we figure out that 2 different genes on different positions. And here, as you can also see in the phenotype of our lines, that those which have YRSLU1 and YRSLU2. The further analysis of the same study, that there are some set of lines which showed resistance against these 4 races of stripe rust. However, they are segregating against this PSTV218. But when we checked with the markers, we figure out that those lines which are actually segregating, they have both genes. That means that these lines could have additional resistance genes. And on other way, our genes, they are also, for sure, they are also controlling resistance against the additional races. So we need to check our material with the additional races too. So the short summary of this article is, we have wheat triintrogression lines, which has 4R, 5R, and 6R Y chromosomes. And we checked with the SSR markers, which are unable to detect the Y chromosome within this SLU 1 to 6 population. We did cytogenetic analysis, and cytogenetic analysis were also unable to detect this short Y fragment. We did GBS analysis, and GBS analysis actually confirms the presence of stripe rust resistance genes. And we figure out that it's a cryptic translocation. And from this study, there is also another study which has these 4R Y chromosomes, and it's still in process. So those set of lines, they are showing resistance because of the 4R chromosomes. So this article has been published, so you can also check. So, and other study is opportunities and challenges where with the combination of novel stem and stripe rust resistance within the same genotype. So the idea was, like in my case, I have this yellow rust resistance genes or stripe rust resistance genes, but my one of senior colleague, he has identified SR59, which is from stem rust resistance genes. So the idea was to combine it within a single wheat genotype. So for this purpose, we did marker-assisted gene pyramiding. So we have a line which has SR59 and YR-SLU resistance genes. So we cross the line which has stripe rust and the line which has stem rust resistance genes. And we did a top crossing with the additional cultivars like here Lingard, Novres, and SLU elite to get the more agronomic performances of our line. And then we did a speed breeding in the normal greenhouse. And since we already developed the cast markers for SR59 as well for our stripe rust resistance genes, this YR-SLU. So we selected the plants by using the cast markers in each generation. And in the later generation, we checked with the several of the stripe rust as well with the stem rust races. So we did the seedling analysis, and we confirmed that we have transferred SR59 plus YR-SLU within a single wheat cultivar. This is a detailed picture of our method. So we have a line which has, wait, let me, I guess. Yeah, okay. So now it's working. So we have a line which has SR59, and we have a line like this at 392, which has YR-SLU. So we crossed in the F1 generation. We did a top cross with an adapted cultivar. It's American cultivar, Lingard. And then in the next generation, when we got this T, we crossed with the adapted cultivar from Tajikistan, Novros. And then in the next generation, that means like in the third generation, this DT, double top cross, we crossed with the SLU, that is a Swedish elite variety. So, and then allow it selfing until 8 or 9 generation. And then we got a variety or a genotype, which has, not a variety, sorry, it's a genotype still. So we got a genotype which has SR59 plus YR-SLU. So when we talk about the speed breeding in the normal greenhouse, so we don't need a fancy setup. In that case, when you are doing this molecular assisted breeding, so we sowed the seed on 21st of April, and on 7th of June, there was a dot development. So after 7th of June, more or less like 1 or 2 weeks, the plants, they were ready to harvest. So we did it in normal greenhouse condition, and the humidity was 40 to 80%. Temperature was 18 to 24 degrees Celsius, but 20 hours photo period was required to speed up the process. And then high pressure sodium lamps, which are normally in the greenhouse, we used. So the main results from this study was that we successfully transferred SR59 and YR-SLU2. The challenging part was YR-SLU1 we lost when we cross it. So from that generation, like you can see from F1 to until F9, we actually lost that gene. That's the challenging part when you are handling with the 2 different diseases within the same genotype. And then the lines also shows a complete resistance to stem rust races, which we have checked in our case. And previously, this SR59 also shows resistance against all of the UG99 races. And then the lines, they were actually segregating against stripe rust races. So the reason could be the absence of these resistance genes, because in because the background line, you can say it was actually strongly resistant, but when we cross it, so it shows segregation with the same race. And then we also confirm the genes of interest in terms of SR59 as well YRSLO2. So we really need to understand what's the mechanism behind the absence of this YRSLO1. So the conclusions of my research is wheat alien introgression line has a distinct potential for wheat improvement. And then wheat rye substitution lines, they possess resistance to 30 races of stripe rust. NLR genes for 4R, 5R, and 6R rye chromosomes are confirmed in our study. And then we validated the Cas markers, which are actually responsible for stripe rust resistance, and which is coming from the 6R long arm rye chromosome within the wheat genome. So this is actually a cryptic translocation which occurred on the long arm, and it is temporarily designated YRSLO genes. And mass gene pyrimidine actually provide the durable disease resistance against stripe and stem rust, apart from the absence or the loss of the gene. So long story short, phenotype and genotype analysis is an effective approach if somebody is looking for the new, for the novel resistance genes. So the future directions of my work are gene designation for stripe rust resistance, which is temporarily designated YRSLO, and then the functional analysis of these novel genes. And we really need to work on with the set of different races, as well additional races from different regions, not only from the Europe, maybe from the Asian races, with the same material. And we need to work on agronomic performances of these races, like diseases and pests. And then end use quality analysis is also required. And the last but not least, it's a gene permitting into a Swedish wheat cultivar. And I hope so that I would be here after maybe 5 or 7 years, since it's a long journey. So I would like to acknowledge Swedish University of Agricultural Sciences, who gave me opportunity to work on this project, especially my supervisor, Professor Eva Johansson, and my co-supervisor, Dr. Mahbub Jan Rahmatullah, and another co-supervisor. She's working in a company, Dr. Pernilla Wallenbach, and I also actually worked at Global Rust Reference Center, and I would like to thank Professor Moines, who gave me opportunity to work there. And thank you for your patience. So if you have any question, you're welcome.
Okay, thank you so much. Everybody clap. We do have questions. Thank you for a great webinar. Okay, our first question from Matthew Bogard. It seems you mainly tested resistance at the seedling stage. Could you confirm the resistance phenotype at the adult stage?
Yeah, actually, the background of this material, like this SLO1 to 6, it also confirms the resistance at the adult stage, but with the 2 to 3 races, not with so many races. Yeah, at University of Minnesota. I mean, we have sent samples there, and my actually co-supervisor Mahbub Jan, he was working there, so he checked these wheat rye introgression lines, and then it also shows the resistance within the field too.
Okay, great. Our next question is addresses the observation on zinc. So higher level of zinc, can you please share the ppm, parts per million?
Actually, the higher level of zinc, as I mentioned before, like my target was with the stripe rust. So I was actually taking care of only the stripe rust part.
Yeah.
Since these are like a large number of lines, so these are the part of different studies. So another PhD student is working with this zinc and then cadmium, but in terms of rye, we got a signal that okay, this is resistance against the stripe rust. So I just continue with the disease, not with the micro macronutrient concentration. But of course, it's a very good concentration. I mean, in the article which we have published, someone they can get from there. I'm sorry for that.
And there was a second part of the question, maybe you know or don't know, but was the zinc, was this, I think if I'm interpreting the question correctly, was the zinc higher at background levels, or did this observation require an application of zinc in order to see the increased zinc levels?
In terms of zinc, I can information which actually I received with the wheat rye interrogation lines. It was the zinc and the iron, they were very good in concentration, and then but with the cadmium it was low. Some lines they were very low, so that's why we actually continue with this set of 6 lines, SLO 1 to 6, and then so on. So I don't remember the exact number, but I can share my publication so they can check their, since it's not my part, because I was actually working on this resistance breeding, because this actually article, this is a review type article, you know, from different countries and different people and different PhD students, they are working on it. So yeah.
Okay, well, back to the topic of your talk. So we have a question from Saif Ullah from the Crop Diseases Research Institute at NARC, Pakistan. And he says that he's curious to know if you've incorporated any Pakistani wheat varieties into your study. Did you encounter any resistance sources during your research? And I'll just finish. Furthermore, do your findings suggest that the identified resistance genes could provide protection against Pakistani strains of rust?
Actually, I'm working on a project. I'm writing application for that, and this could be, of course, I would like to collaborate with Pakistan for sure, but at the moment it's still in process. But if I talk about the background of these lines, so these are not checked with any of the Pakistani races, not yet.
Okay, here's another question. Did you have CASP markers for both YR genes from the SLU 126? Why then did you lose the resistance? Can it be due to segregating suppressor genes in the backcross parents?
Yeah, it's quite interesting question. That's what you're actually looking for. But I guess it's, I mean, on both sides, like when we are crossing with this Chinese spring PH1B mutant, so that could be the reason that we lost this YR SLU1, or that could be a reason that when we are working with two different resistance genes from 2 different diseases. So this could be a reason that we lost it. But his question is about like, well, since we have the CASP markers, yes, we have developed the CASP markers, but we really want to transfer within the single wheat genotype. So that was the idea.
So I just trying to clarify in my mind. So you had 2 CASP markers, and they detected the same integration event, but there are perhaps multiple genes in the introgressed region. Is that?
Yeah. They are, I mean, two different genes on the different positions, but
Okay.
the origin is same, like from 6 or right chromosomes, the origin is same.
So you may be able to find out from the segregating population if you discover more markers, you can potentially look at whether there are other genes that are required.
Yeah, of course. But yeah.
Okay.
Yeah.
Okay, let's move on. We have a question. Rye seems fine, but did you check for any potential linkage drag?
Sorry, can you repeat the question?
So I think, I mean, part of the purpose of your research, you have these large substitution lines, and then you're trying to generate smaller integressions
Yeah.
in order to eliminate potential linkage drag. And I guess the question is, have you actually looked at linkage drag, and have you been able to see that there is or is not any?
No, I did not look at it, but of course there are studies like when you just find out that it's a cryptic translocation, then of course there are some studies about this linkage drag. So, but I did not look at it. But maybe that could be a part of further analysis. Yeah.
Okay.
Yeah.
Next question. We have lots of questions. Was there any assessment of possible pleiotropic effects when combining SR and YR resistance genes?
In terms of our study, if I just talk about our study, I guess we got very good agronomic performances when we grow them within the field here in Sweden. So like flowering, maturity, seed color, hardiness, and I mean all of the characteristics, they were really good. Apart from that, this material which has this SR59 and YRSLO2, so this material actually shows resistance with the Fusarium head blight, septoria, so additional diseases too. So yeah, I can say that
there might be kind of interactions.
Yeah, like a little bit, yeah.
Okay,
But still, of course, I mean, we need to work on it. It requires a lot of money and a lot of time.
Okay, and I think this next question kind of relates to the linkage drag question. So what are the disadvantage of transfer gene on quality and overall acceptance of genotypes?
Okay, good question. Yeah. In our case, as I mentioned before, the lines which has this wheat-rye introgression, those set of lines, they are very good in agronomic way, as well with the disease, as well with the end-use quality, and concentration is also good. So that was a reason to continue with these set of lines. So the question is particularly about, well, like when you are working within the tertiary gene pool, like since rye, we are getting the gene from the rye. So I don't think so that in our case it would affect. No, it's not affecting as per our analysis.
Okay.
So it's good. Yeah.
Okay, this next question has to do with understanding the methodology. This attendee is asking for a clarification on the CSPH1 mutant genotype. So how was that mutant genotype developed? I mean, I don't know. Is there a concern? Yeah.
Mutant? Actually, okay, it was not developed. It was, I really acknowledge one of the professor at University of Minnesota. He actually gave this material to my co-supervisor, so he just continued with it. Yeah, so it was not developed by himself, no.
Yeah, so this is a useful line that can be used, that has been used historically. Is that correct? Is that the same?
Of course, of course.
Okay. Next question. Would you advise on effective strategies, methodologies for combining resistance genes for the 3 rusts in wheat, a single genotype? So
interesting. Yeah, 3 rust.
Yeah.
I don't have any experience with the leaf rust, but of course have experience with the stripe rust as well, with the stem rust. for me, if you got a gene and then you develop a marker, like in case of SR59, this shows a strong resistance with so far all of the UG99 races. However, in our case, for stripe rust, these lines shows resistance maybe 30 or more than 30 races. So the first strategy is like you really need to check the material for agronomic performances, like how this material is behaving within the greenhouse as well in the field by using different parameters. And then secondly, the gene which that material has, it should be more or less like how we can say like a fixed, like in case of SR59, because I knew the nature of this YRSLO1. It was also switch on and switch off in my study throughout from F1 to until BC2 F4 generation. So it has a higher chance, like when you're checking with the markers. So if the gene is there, I mean, it's constantly showing that it is there, then it means like, don't need to worry. But if it's switch on and switch off within the same background, then higher chances to lose it. So that was the reason that when we cross it, so in the F1 generation, you just lost it. So it really depends on the gene of interest too.
Okay. I'm okay. So there's a technical question for the GBS alignment to the rye genome. Which one did you use? I'm not sure if that's referring to the rye reference, and are there multiple references that you might have used?
Okay, actually, we send our material to the company. So they send us the sequence, and then we choose and then we actually selected the high quality SNP. So we have a bioinformatician there in our university as well. We have collaborated with IPK and joining us in the UK. And then after that, we developed the cast markers by using some softwares and then apply it. So in the beginning, we actually aligned that sequence with the rye as well with the wheat. The reason, because we were looking for rye chromosome within the wheat genome. So that was actually the strategy.
Okay. Is it reliable to evaluate disease response on mere phenotypic basis? Or are there any biochemical tests or PR marker, genes expression analysis, histopathology, et cetera? So for your studies, are you generally just looking at the disease response, or are there other assays that you may perform that could also inform resistance?
I mean, during my PhD, I was just looking at the disease side, but of course now I really want to, as I mentioned in the future directions, that I really want to see the function of these genes. Are these only for the stripe rust, or maybe these genes could control the other diseases too, resistance against other diseases too. So yeah, so far, yes, for the additional diseases and then functional analysis. Yeah, but let's see.
Okay. Do you have any yellow rust races from East Africa that you might be testing?
Actually, we are working at GRRC, Global Rust Reference Center in Denmark. So for sure, they have all of the races, since it's a super big one here in Europe. So, but I did not test it with the East African race. But I guess since they have some codes on the races when we checked. So, but of course, with the African races, I have checked with with my material against the African races, but I'm not sure which one. I mean, it's the East African or from which side. Yeah.
Okay. This question has been upvoted several times, so I'm going to read it. This is how does the integration of novel YR and SR genes into wheat breeding programs impact yield and overall plant health? What are the current challenges in identifying YR and SR resistance genes in wheat, and how does your research address these challenges? So multiple questions.
I guess the first part I already answered in terms of like challenges, and I can also explain a little bit more. Like when you have a very small translocation, so there are higher chances to lose that gene when you are working with this gene permitting. And regarding the second part of the question about my research, more or less, I remember that I spent 3 years to identify these triplast resistance genes because the SSR marker, they were not working. But of course, we need to work on the method. I mean, at that time, maybe the only reason was funding, I can say. Otherwise, we can check in each generation with the status by using the cytogenetic analysis, but we could not do it at that time. We were checking with the SSR and it was switch on and switch off. I guess in terms of improvement on my regarding my research, I really need to think about the alternative methods too. If that method is not working, so you have to work in that way. Yeah. So since I have identified the resistance genes which are showing resistance more or less more than even though 30 YRC, so I think so it really plays a significant role. And I also have developed a CASP markers which are really good, and farmers and breeders, they can get that sequence and then they can also use it. So,
well, but would you say that the breakthrough here for you was using GBS in order to increase the number of markers you can assay?
I mean, no, I mean, with the GBS, it was okay. But in the beginning, when we use SSR markers, simple sequence repeat markers, so it was really time consuming in the beginning.
Right.
So
would you
yeah.
continue to use GBS if you were to?
Yeah, exactly. And
okay.
Then yeah, exactly. And then we got the picture, the clear picture. So right. I would consider that the GBS is for sure. It's the most smarter way when you are looking for resistance genes and identifying and developing that markers.
And I think in your paper, did you say that the integration was like 5 megabases? Is that true?
Sorry?
The integration in SLU on chromosome 6, was that like a 5 megabase integration?
Yeah, exactly. I mean, you're talking about the size of the size of the integration.
Yeah. Right.
Yeah, exactly. That was the reason that we were unable to detect with the cytogenetic analysis.
Mm-hmm. Okay.
Because the cytogenetic analysis, it requires a little bit longer stretch of this transfer.
Okay. We're starting to run low on time. We have about 2 minutes left and about 8 or so questions. As we stated before, Rimsha has kindly offered to answer these questions offline in writing. So I think this might be a good time to stop and thank once again everyone for their attendance at this very interesting webinar, and to thank Rimsha again for really a terrific and informative webinar on rust diseases. Thank you so much.
Thank you. Thank you so much.